I hope I am audible. So today's session is on composition of blood. So let's have a look at what all will be there in your blood. Okay, so we all have, all the animals, when we talk of mainly, so we are all having blood. Okay, so but again there is a variation with respect to blood, with respect to the pigmentation. And some animals, when we mention, in case of a starfish, so we say that there is a water. Okay, so water is the one which is circulating. So it's not that always all the animals will have a blood. Okay, so but most of the animals will have a body fluid as a blood, and some of them will have a water, in case of starfish, then there is porifera, where the circulating fluid is again a water itself. Okay, so let's have a look at the blood today, which is a body fluid. So it's not, it is not just the blood which is present in our body. There is a tissue fluid, there is a lymph. So we will study that as well. Just a second, let me just check this. It is not just the blood which okay is all right. Okay, so let's get started with this. So before I start, let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So you can join with us in Telegram. So for homework and class notes, you can join the channel Vani Kari ICSE class 9 and 10, and Telegram channel, that is Unacademy ICSE and ISC both, you can join. And this particular link you will find in the description box. Okay, so you can go to the description box and click on this particular link, so that you will be joining the Telegram channel. Also, we are conducting special classes on Unacademy platform. So all you need to do is download the Unacademy Learner app and start using it, so that we are conducting one shot revision, we are conducting the poll war, like a quiz we are conducting. Then we are also conducting the preparation strategies or the time management. So a dual session is run by two of the educators. So this is already done. Okay, so yes, you can watch the recordings which will be available here. So this is gonna help you in preparation for your finals. Okay, so let's see what happens in special classes. So special classes are basically free classes. So live real time interaction using the chat and the emojis. Okay, so basically you have a chat option which is available here in YouTube. So same way, there is a chat option which is available, the emojis are also available. And there is a chance that when lot of questions are lot of comments are coming from the number of students. Okay, so there is a chance that your question might get lost. So let's say there is an interaction session going on between an educator and a learner. So when you are interacting, so you might have certain questions to ask, or discuss with the educator or other learners as well. So in that case, we have a separate question tab. Okay, where you can type in your question, or also there is a option to attach an image, and you can send it to the educator during a live lesson, a live class itself. Okay, and they'll be getting a notification that the learner has asked the doubt. So they'll be clearing your doubts during a live class itself. Also again, like I already said, so there is a quiz which we can conduct, or a poll what. So basically you will get options whenever we give a poll. So you will get these options to choose between. So the one which is correct you can choose. Okay, so there is a poll war that we are conducting. The question and the options will be available on a screen. Okay, then there will be a pop-up, poll you will be getting, where you can choose these the answer. Then so after the quiz is done, so we are also sharing the leaderboard. So with the leaderboard, you can know your rank. So this is gonna encourage you to perform more or perform better, or so basically you will have a competition. So compete with your friends, so that you can know. So this is basically with respect to the leaderboard. Okay, and Unacademy subscription feature. So this one, until now here I was mentioning about the free classes. Just a second. Okay. Okay, so until now I was mentioning about the free classes which are conducted by us. Okay, and here just second, something is wrong, I guess. Yeah. All right, so this is about the plus subscription that we are gonna conduct. So this is basically the plus classes or the paid classes these are. So what is the advantage or what is the difference is that, so one thing is anyway you can learn live from the comfort of your home. Okay, unlimited access to all the courses. So there are different educators who are conducting different they will be conducting a different courses. So now we have come up with lot of crash courses for you all on different subjects. Okay, so for all this chapters again, so you will have access to all the different courses run by different educators if you are going for a subscription. Okay, also with that, the top educators of India are going to take the classes on one platform, so that is Unacademy platform. Then regular doubt clearing session will be done. So like after about every 3 sessions, so we are conducting one session, the fourth session, which we call as a doubt clearing session. So any doubts you have related to the topics that are previously covered, so you can clear your doubts in this a specific session. So this is completely meant for learners itself. So any doubt they have related to the topics, they can clear their doubt. So let's say they do not have any doubts, so the learners are not having any doubt, so we will be revising the topics that are previously done. Or if the learner expect, like if the learner wants to conduct a practice session, like if they want us to conduct a poll war or a quiz, what we call, so that can also be done in this particular session. So basically answer writing session also, which we call. Okay, then complete coverage of a syllabus is done, then mentorship and guidance is given, study material is given, practice test series, live test series. So all these test series you can attend, and the batches. Okay, so if you prefer to learn in Hindi language, so there is a different batch. If you prefer to learn in English language, then there is a different batch. So you will have access to both of these batches, so you can choose your preferred batch and go for that particular batch to learn from a educator. Also with that, so there is a daily practice section, weekly mock test series, live quizzes, daily MCQ and subjective test series. So you can have a benefit of all this with the subscription. And if you are planning to go for a subscription, so if you want to try like how exactly this works, so you can try with the 3 months or maybe 6 months. Okay, and if you are planning to complete your class 11, 12, so maybe you can go for higher months of subscription. Okay, so this is again completely depending on your convenience and your plan. So you can go for any of these months of subscription with the referral code VANIYT, because today there is an offer of 20% discount. Okay, so if you are going for this particular offer, okay, only today the 20% off is there, or otherwise it will be usually 10% discount will be available with the same code. Okay, so also unlock 20, so this is the one, and you can use the code VANIYT. Okay, so VANIYT is the code where you can use to get 20% discount on the plus subscription. So this is mainly, one second, yeah, so this is mainly for the plus subscription itself. All right, and also there is a mega offer. So offer for plus subscription, that is for ICSE class 9 and 10 and ISC 11 and 12 also. So let's say you are going for 12 months of subscription or more also, so basically you will be getting free 2 months extension. That means 14 months of subscription you are getting with 12 months. Okay, so flat 20% off today. Okay, with the referral code VANIYT. Okay, so this is my referral code. Vani is my name, YT for YouTube. So VANIYT is the referral code. You are going to get 20% today. So this is valid for the first 500 subscribers only, so you can go for it today. And if you are having any queries with respect to preparation journey, or about the subscription or about the latest offer, so you are just one call away. So this is, you can call on 8585858585 and use the code. Don't forget to use the code, that is Vani YT, because with the code itself you will be getting a discount. Okay, so let's get started with the topic. Okay, so composition of blood. Okay, so composition of blood, when we mention, you know, what all comes to our brain, basically what all comes in our mind. So basically it is the a plasma content which is there, a liquid part is there, and there are cells. So mainly we think of red blood cell. And again, when we mention when we think of a blood, so we imagine is a red color. Okay, and red coloration is because of what? So that is because of the RBC, which is having the hemoglobin pigment. Okay, so let's have a look. So what exactly we are going to study? So introduction related to the blood, then we will see what is the blood, then what is plasma, the formed element or the cellular elements also we can mention. Okay, and those cellular elements are what? That is erythrocyte, RBC. Okay, leukocyte, that is WBC, then thrombocyte, that is platelet. Okay, so we will see each of these today. Okay, in this session we will study what exactly are this. All right, so now the blood. Okay, so at the beginning of your chapter circulatory system itself, so you have noticed that the function of blood, when they mention the blood is there to circulate throughout our body. What is circulating? What is it circulating? So it is the nutrients, it is the oxygen. What would happen if blood is not there in our body? What would have happened? Like, you know, if the blood is not there, oxygen we are taking in, how exactly oxygen is going to your tissue? How exactly, you know, we are taking in a food now. Okay, so digestion is occurring. So there is a simple compound. So you know, digestion, when we mention, complex food is broken down into simple one. Now this simple food, where it is going? It is going to the blood, and from blood only it is going to the tissue. So if blood is not there, what will happen? There is no oxygen supply, there is no nutrient supply. So same with respect to your endocrine system, because hormones are being supplied through the blood itself. So there is no hormone supply, there is no waste product removal. What will happen if the waste product is not removed? All the toxic compounds are going to accumulate in our body. So that is again dangerous, right? So that is why, you know, blood is so important that most of the functions which are occurring in our body, so this is all happening because this blood or a fluid which is there, so it is circulating throughout the body. Okay, and you know, each and every cell or each and every tissue is getting a nutrition. So what exactly is happening? By anyway, yes, blood is supplying nutrition, we are saying, and blood is supplying oxygen, we are saying, all of this. Okay, but what exactly is the use? So you know that, right? The cellular respiration. So now this is a cell. Okay, so this cell is supplied with the oxygen. Okay, this cell is supplied with the glucose now, and you know that the cell is having a mitochondria, and mitochondria, what do we call it? It is a powerhouse of a cell. So what exactly is happening here? This glucose is getting oxidized, and there is a formation of carbon dioxide, a water, and then you know the main component is ATP. Okay, so ATP is generated. Why do we need this energy currency? So this is the energy currency. So why exactly do we do it? I mean, why do we require it? So that is to perform most of the body activities. You know, I am able to talk to you all, I am able to sit in this position, you know, I am able to run, you are able to run, you are able to dance, you are able to perform most of the activities, your brain is working well. Okay, so you are able to read, you are able to concentrate. So most of the activities that you perform, so for all of this, you know, and yes, main thing, you are growing. Okay, so yeah, this is the age where you are growing, right? So growth is occurring because cell division is occurring. So for cell division to occur, ATP is required. So lots of ATPs are required for a cell to divide. Okay. So this is all happening with respect to this transport system, which is supplying all the necessary elements to the tissues, and in the tissue I mean mainly the cell, where the mitochondria, where the ATP synthesis, the powerhouse is where the energy, okay, that is ATP is generated. Okay, I hope you got the point. So this is with respect to circulating fluid. Okay, so different organisms are having a different method. So now in our case we are doing it by a blood. Okay, but different organism, like the starfish, like we are sponges, cylindrics, so there the water is the one which is supplying. So through the water only they'll take oxygen, through the water itself they'll get rid of the carbon dioxide if it is there. So through the water itself the waste products are being removed, through the water itself the nourishment is taken. Okay, so all this can be done through the water in these sponges and the cylindrics, starfish. Okay, so that is the case. Now higher organisms including the humans, so they use the blood and other body fluids. So we use blood and other body fluids, so that is the lymph. So let's have a look at what exactly is there in the blood. Okay, so blood, when we mention, so this is a fluid connective tissue. Okay, so fluid connective tissue or a specialized connective tissue which will have matrix. What exactly is matrix? Okay, so matrix is nothing but the plasma, a liquid part, and this plasma is the one which is having most of the required protein, the water content, the salt, glucose, and the digested products. So we said that digestion is occurring, complex food is broken down into simple ones. So where exactly this is going? The simple food is reaching the what we call as a plasma. So in the plasma only all this simple food that we can find. So comprises fluid matrix, that is plasma, and formed element. So what exactly is formed element here? So this is the cellular element. So cellular element is all the different cells that you find. So now plasma and cellular element, when we mention, so 55% of the blood, what is there? So that is completely plasma, and 45% of the blood, what we mentioned, so that is completely a formed element or a cellular element. So let's have a look. So like what exactly is plasma, like what exactly it contains, and what about the cellular elements as well. So minerals, when we mention, there is a sodium, there is calcium, there is magnesium. Okay, so all the different minerals that you are consuming, that you are taking in, you drink a tender coconut, so you are getting lot of minerals. Okay, so basically it is it is a nucleus itself, which is a free nucleus which is there. Okay, so that is how the coconut is having a water inside, because cells are not formed after the division. It is a free nuclear division, so it is basically a nucleus. Okay, so yes, we will talk about that later basically. So now the water which is there, so this is rich in sodium, this is rich in calcium, then the magnesium. So this can be with respect to plasma. When you are taking a food, they are going to enter your blood part, that is the plasma part. Then this is again with respect to digestion. So amino acid is a breakdown product of a protein, so that you are getting into the plasma. Glucose is a breakdown product of a carbohydrate, so that you are taking into the plasma, again through the absorption process that occurs at the small intestine in the villi. Okay, then lipids also, it is the same thing. So fatty acids, okay, so fatty acids and the glycerols are the these are the simple products or the breakdown products of the fats or lipids. So these two are also can be there. Again from here these can be supplied to the respective tissues. Okay, so 6 to 8% protein is there. So there is a fibrinogen. So this fibrinogen is responsible. I hope this is visible. Let me just check. Okay, so next time now I'll make it little bigger. Okay, So this slide I'll make a little bigger, or the font size I'll increase so that you can clearly see. Okay, so this one is fibrinogen, which is there. So fibrinogen is there, it is having a role in clotting. Okay, globulins are a proteins again, so they are having a role in defense mechanism. So what exactly is globulin? So it is the antibody. Okay, so antibodies are having a role in defense mechanism. Then we have albumin. Okay, so albumin is basically there for the osmoregulation. So you have studied osmoregulation function in the excretory system. Okay, so osmoregulation of a blood will occur. That means whatever normal concentration of a blood has to be maintained after the ultrafiltration process. So reabsorption of water will occur, reabsorption of glucose will occur. So maintenance of osmotic balance or osmotic pressure of a blood, so that is done because of this albumin protein which is there in the plasma part. Okay, so then there is, most of it is again a water, that is 90 to 92% is a water. Okay, so this is about the plasma. And when we have a look at the plasma, so this is straw colored. Okay, so straw is basically the somewhat yellowish color you can find. Okay, so that is dry plant. When you observe, it is yellow color, right? So that is the same one, straw, which we mentioned. Okay, so it is a viscous fluid. Why it has got a viscosity? It is because of all these compounds which are present. Okay, so there are proteins present, there are amino acids present, these are minerals, glucose, lipids. Okay, because of all this fluid has got the viscosity, viscous fluid it is. Then 55% of the blood volume is again a plasma, and also it has the clotting factors which is there for a coagulation. Okay, so moving on to the next part. So there are formed elements. I cannot see your chat. Okay, so if I had, I don't know why the chat option is anyway not available. So you can anyway get in touch on Telegram channel. All right, so formed elements, so this one formed elements are platelets, leukocytes, erythrocyte. So erythrocyte is RBC, leukocyte is WBC, platelet it is a platelet or a thrombocyte. Okay, so if we are taking mm cube of a area, okay, so let's say this is a mm cube of area now. Okay, so only in this mm cube, so platelet if we are finding, so we can find up to 1.5 to 3.5 lakhs of platelet we can find. And if we are talking about the same area, so let's say this is the mm cube area now, so 6,000 to 8,000 WBCs you can find in here. So let's say this is the same mm cube area, so you can find 5 to 5.5 million. So which is the most abundant one? RBC is the most abundant one. Okay, that means many millions of cells are there. Next comes the platelets. So RBC you can find in millions, platelets you will find in lakhs. Okay, so I'll just write it here. So this you will find in millions, this you will find in lakhs, so this you will find in thousands. Okay, so you can see that this is the most abundant one. Next comes the platelet, and then there is a leukocyte. So these are the structures. So this is a platelet, and we will see why exactly, based on what we will make a difference between these structures. So it is mainly based on the nucleus. Okay, so all these are WBC. So all this is which is mentioned here, so this is all WBC, this is platelet, and this one is the RBC. Okay, and we will make out the differences between these WBC by looking at the nucleus. So you can see that the basophil nucleus is indistinct. Okay, so it is one time it is like this, so it can become something like this, it can become something like this, so it is indistinct, so we cannot clearly make out the lobes which are present. Then eosinophil, when we mention, so eosinophil is having a headphone shaped nucleus. Okay, so this is perfectly like this, and it is always like this in case of a eosinophil. So based on the nucleus only, it will make out a difference between these WBCs. Lymphocyte, you can observe here, so there is very less space available for a cytoplasm. Most of the area is covered by a nucleus itself. itself. Okay, so this one is a lymphocyte. Then neutrophil, you can see 3 lobes here. Okay, so this is a 3 lobe here. It's not that it will have only 3 lobes. Okay, so there can be 4 lobes also. So this neutrophil, when we mention, so this is polymorphonuclear neutrophil. Okay, or a polymorphonuclear cell also we call for a neutrophil, because many lobes of a nucleus will be given. Yes, so why I am saying this one is, in your exam, so they'll give you a neutrophil, or they can give you a monocyte, they can give you a lymphocyte. So they'll ask you to recognize, or they'll ask you to identify and write what exactly it is. So which type of a WBC they'll ask. So if you write WBC, then you might go wrong, because they are specifically asking what is the type of WBC. So you need to mention whether it is a granulocyte or whether it is a granulocyte. Okay, so you need to be very sure about that. So whether it is a granulocyte or a granulocyte. Okay, and also with respect to their structure of a nucleus itself, you need to make out the differences. Now neutrophil is polymorphonuclear, but monocyte, when we mention, so this is having a kidney shaped nucleus. Okay, so kidney shaped nucleus if you find, so that is a monocyte. All right, so let's get with the next one. So composition we saw. So plasma, we saw that it is forming about 55 to 60%. Cellular is about 40 to 45%. So water is this much, protein is this much. So there are inorganic traces. Then other substances can be glucose, amino acids, then the fatty acids and the glycerol. Okay, so again with respect to inorganic salts, so it can be sodium chloride, NaCl, sodium bicarbonate is there, and another substances contained in the plasma. So it can be glucose, amino acids, the protein is fibrinogen, hormones can be there, urea is a waste product. Okay, so this also forms the compound. So this one is a waste product which we need to get rid of, that is through the kidney. Okay, in the form of a urine. So that is also there. That filtration. Okay, so what is happening, the ultrafiltration is filtration process is happening at the kidneys itself, and that is how we are getting rid of the unwanted compounds. So this has been removed. Okay, so the plasma. Okay, so when the plasma is there, so from the plasma, so that the plasma will have a fibrinogen. But if we remove a fibrinogen from a plasma, okay, so when we remove this fibrinogen from plasma, then in that case we call it as a serum, and serum does not clot. Okay, why it does not clot? Because the fibrinogen is the one which is responsible for clotting, and if we are removing a fibrinogen itself, then this serum is not going to clot. Okay, so there is no clot formation. That is why a plasma from which a protein fibrinogen has been removed, so that we call as a serum. Okay, so let's have a look at the cellular elements. So there is a RBC, leukocyte, WBC and thrombocyte. So these are the platelets. Okay, so these are the formed elements. RBC. Okay, so this is for a erythrocyte or a RBC. So RBCs are most abundant one. What is the number? 5 to 5.5 million per mm cube. Okay, so this was per mm cube. So this is the most abundant one. Where is it formed? It is formed in the red bone marrow. Okay, so this is devoid of the nucleus. What is the meaning of this one? So let's say this is a RBC now. Okay, and you will find RBC is disc shaped. So now this one is the top view. Okay, so this is, we are viewing it from the top view. So if we observe it from the side, so this appears like this. Okay, so this appears as a concave structure. So by concave structure, so that is mainly, here the nucleus which is supposed to be there, so that nucleus is absent, so that is why it has taken the shape of a disc like this. Okay, so this is the side view. Okay, so this is a side view, this one is a top view. All right, so here in this case, so devoid of nucleus in most mammals, It is biconcave because of loss of nucleus, as the nucleus is not there, so if this is the surface, very less space is available, but if the nucleus is gone, so this will become like this. So nucleus is gone, so this was the condition earlier. Okay, and if the nucleus is gone, what will happen? So this space is available, so as there is a groove and ridges formation will occur, so more surface area is available. If more surface area is available, what is the advantage? The RBC can carry oxygen. Okay, so it can carry more oxygen. So now when we mention that the RBC is devoid of a nucleus, so there is no nucleus, so that surface area is more. Okay, so I'll just write this. Okay, so surface area is more. Now mitochondria is absent. Okay, so when mitochondria is absent, what is the benefit? Mitochondria is one such organelle where the cellular respiration occurs. Okay, so now here in this case, as mitochondria itself is absent, so RBC is not going to use the oxygen for itself, so oxygen usage is not done, so that it can supply oxygen to different tissues. Okay, so of different tissues are going to get the oxygen because the RBC itself is not using oxygen. So this is another usage. Okay, and other than that, there is endoplasmic reticulum also which is absent. So what is the benefit of this one? So if endoplasmic reticulum is absent, so flexibility is provided because the RBC has to pass through the narrow capillaries. Okay, and it requires a flexibility, a little of a flexibility is required, so that it can it can pass through these narrow capillaries also. So that is why, so absence of this endoplasmic reticulum will provide a flexibility, as endoplasmic reticulum is responsible for providing a rigidity or a particular structure to the cell. So when it is absent, a flexibility is provided. Okay, so lifespan, again you will get questions on this. So 120 days is the lifespan. So after this is done, after it has completed 120 days, so RBCs are gonna die in the spleen. Okay, so that means the spleen is where the degradation of the RBC is gonna occur. So the hemoglobin is gonna get separated and the RBC, that is the cell, is gonna get separated, and this hemoglobin, when it is broken down, okay, when the breakdown of a hemoglobin occurs, so that is when the formation of bile pigments will occur. So bile juice formation, so bile pigment formation is basically breakdown product of a hemoglobin. Okay, so that is how these bile pigments are produced. Okay, so here we were discussing about the erythrocytes. So these are red in color because of the hemoglobin. This is biconcave. So other than that, this is the size. So 7 micron in diameter. Okay, so that is 7 micrometer. Okay, so 7 micrometer is the one. All right, so then other than that, the small size, that is basically the concavities because of the larger surface area, as the oxygen is absent, so it is very efficient in absorbing the oxygen. I hope you all are getting. Yes, are you all understanding? So since chat is not available, I am not getting a chat option here, the chat is not visible, so I hope you are all getting. So if you want me to explain something, maybe you can get in touch with me on Telegram as well. Okay, alright, so the small size enables the red blood cells to travel through the very fine capillaries. Okay, so that is how the endoplasmic reticulum absence is gonna help. So now adult human male, okay, so male is having 5 million RBC and female is having 4.5 million RBCs. So hemoglobin, hemoglobin is, it is having globin protein is there. Okay, so there is a protein part. So hemoglobin is basically, globin is for a protein part. Okay, and then heme is for a iron group. Okay, so there is a iron group which is present. So RBC contain a red colored iron protein. Okay, so this one is a iron protein. Okay, so amount of hemoglobin is gonna vary. So this is, in case of adult, it is 12 to 16 gram per 100 ml of a blood. Okay, so transport is basically the transport of the gases, the oxygen and the carbon dioxide. So these are being transported by the hemoglobin itself. Okay, so hemoglobin, so there is a hemin part, that is the iron group, then protein part is there. Okay, so let's have a look here. So when the oxygen combines with the hemoglobin, so the product formed is oxyhemoglobin. Okay, so this oxyhemoglobin which is there, so this is a temporary product, or we can say it is an unstable product. Okay, and whenever there is a necessity of oxygen to the tissue, so this oxyhemoglobin, the hemoglobin is going to get separated from the oxygen and oxygen supply will be done. Okay, so same with respect to carbon dioxide. So carbon dioxide combines with the hemoglobin, forming the carbaminohemoglobin. So there is a formation of a carbamino. Okay, lot of students I have seen, so they will write carboxyhemoglobin. Okay, so carboxyhemoglobin formation is for carbon monoxide. Okay, carbaminohemoglobin formation is for a carbon dioxide. So be careful when you are writing this one. Okay, so carbon dioxide is carbaminohemoglobin. Again, this one is a unstable one. So we need to whenever there is a carbon dioxide that has to be thrown out of the body, that is by exhalation, so this carbon dioxide will get separated from the hemoglobin. So this is also unstable compound, but with carbon monoxide, so this is not possible, because so the carbon monoxide will form a stable compound. That means carbon monoxide plus hemoglobin will form a carboxyhemoglobin complex, and we cannot separate this two. So this when the hemoglobin count is going to decrease this way itself if there is a carbon monoxide poisoning. So if more of a carbon monoxide is there, so then stable compound formation will occur. So stable compound is carboxyhemoglobin, and that is how one needs to get replaced. New hemoglobin formation has to occur there. Okay, so moving on to leukocytes. Okay, so white blood cells, so these are nucleated. You saw that RBCs are enucleated. They do not have a nucleus, but white blood cells, so they have lot of different types of nucleuses are there. So it is a nucleated, it is short lived. Okay, so it can survive for hours also, and depending on the infection, the WBCs can survive for years also if there is an infection, or otherwise it is basically for certain hours, or we can say in days, like 10 days we can mention here. Okay, so then two categories are there, granulocytes and agranulocytes. Okay, so here WBC, so there is a leukocyte which we can mention, so they are present in thousands. Okay, and they can squeeze out through the capillary. You can see here, now this one is a capillary. You can see pseudopodia formation is occurring. So through the capillary they are squeezing out, they are moving out where there is a bacteria, where there is a pathogen, and this process where through the capillary, where it is moving across the capillary or squeezing across the capillary, so that process we call as a diapedesis. Okay, so dia is across, pedesis is oozing out, so it is moving across the capillaries there. Now based on shape and other characteristics, okay, so white blood cells are classified into two major categories there. Okay, so that is granular and agranular. So let's have a look at what is it actually. Okay, so here the granulocytes, so there are three types are there: neutrophil, eosinophil, basophil. So this is depending on whenever we want to observe them under microscope, so they will take up the different dyes. dyes. Okay, so not all the dyes are taken by all the cells. So this one will take a acidic dye. Okay, so this is gonna take the basic dye. You know, the dye is basically coloration for observing under a microscope, and this is gonna take up the neutral dye. So based on that, the names are given as neutrophil, eosinophil. So we can also mention eosinophil as a acidophil also. Okay, so eosinophil and then a basophil. So these are the granulocytes. So why do we call a granulocyte? Because in the cytoplasm, so they have a granules. Okay, so granules are there. So these are basically the granules are, so they can have the enzymes. Okay, so they can have stored food also. So these enzymes can act on the pathogens, they can kill the pathogens. Okay, so that is why the granules are having an enzyme. Then a granulocyte, when we mention, so cytoplasm of these cells, say they do not have a enzyme, so there are no enzymes in this case. Okay, so let's have a look at this one. Okay, so granular and non-granular. Okay, so granular contains the granules. So nucleus, it is basically having a lobe. So you can see neutrophil, neutral dye, eosinophil, acidic dye, basophil, basic dye. So you can see headphone shaped nucleus here. Okay, and this one is a many lobed nucleus are there, and here this one is a indistinct nucleus, so we cannot clearly say how many lobes are there. And you know the nucleus, so this is with respect to the same one. Okay, so this I already mentioned. So cytoplasmic are granule, it will take up acidic dye. So this will take up the basic dye, the methylene blue. Okay, then nucleus is large, indistinctly lobed. All right, so then engulf the bacteria, phagocytosis, where it is produced? It is produced in the bone marrow. Then this one, this also engulfs the bacteria, produces antitoxin. What is antitoxin? It is basically a antibody itself, or a protein which is secreted by the WBC to fight off a pathogen. Okay, and it is having a role in case of allergy also. One second. Yeah, and it is again produced in the bone marrow. Yes, I'll just say about this. So you know this is a bone marrow. Okay, so here is the bone. All right, so there is a yellow bone marrow over here. So this one is a yellow bone marrow, and here you can find, so at this spots. Okay, so in sponges, I mean this is a spongy tissue. Okay, so that is spongy type of a bone will be there, and there you can find the red bone marrow. So red bone marrow is where the RBCs are produced, yellow bone marrow is where the WBCs are produced. So bone marrow is where the WBCs, that is at the yellow bone marrow, WBCs are synthesized. Okay, so one second. Yeah, this one here is a yellow bone marrow. All right, so moving on to the next part here. Okay, as you can see, so non-granular, so they do not have a granule. So lymphocytes, so single large nucleus you can find. So lymphocytes, so you need to know the percentage of each of these. Okay, so now you know very less cytoplasm is there, but a very large, like you know, big large nucleus is present. So smallest of WBCs, single large nucleus. So this produces the antibodies. Then bone marrow and lymph, where it is produced. So basically bone marrow is where the production occurs. The maturation of this cell is going to occur at the different parts. Okay, so monocytes, so this is forming the concentration of, like you know, 5 to 3%. So 5.3% is the concentration which is there in case of a blood. And other, you know, nucleus is large, it is kidney shaped. And in infection, so whenever there is a infection, okay, so that means whenever a pathogen enters, so this monocyte will it will develop into a macrophage, and once it develops into a macrophage, so it will perform the phagocytosis function. So phagocytosis is ingest the germ. Okay, so that is done by the WBC. So what exactly is the function of WBC? So that is to phagocytose. Okay, engulfing. Okay, so it goes on eating all the pathogens that are entering. Okay, so as the engulfing is occurring, okay, So that means as the phagocytosis occurs, so these pathogens are digested inside the WBC itself. All right. Inflammation, whenever there is a damage in your skin or any parts, so you can see rush of these WBCs there because pathogen entry will be there. So that is where we can find a swelling. So inflammation occurs due to reaction of tissues to the injury. So whenever there is an injury, so usually you find a swelling around that. So that is basically, WBCs are rushing, germs are entering, the germs are entering here. So that is why the WBCs are rushing. So all of them will accumulate there itself. Okay. And because of that accumulation, so we can find a swelling. Okay. So inflammation is basically a swelling itself. Okay. So the inflamed spot has several characteristics, that is increased local heat, redness, swelling, pain. So here the leukocytes, so these are basically especially the monocytes and neutrophils, so they will migrate. That means diapedesis is gonna occur. So they'll move across the capillary and destroy the germs which are there. So they'll also destroy the damaged cells by a phagocytosis. So here the tissue damage is there. So here a group of cells which are there. So all these tissues are damaged. Okay. And WBC is coming to engulf the damaged tissue also. WBC is coming to engulf the pathogen also. Okay. So that is what is the function of a WBC. Okay. So formation of antibodies, that is, the proteins are produced. So proteins are basically, they are defense, they are having a role in defense mechanism, where these antibodies are, proteins are basically antibodies and antibodies are protein in nature. So these antibodies are gonna bind with the antigen. So there is an antigen and antibody reaction which will occur, and because of which, so there is actually formation of this antigen antibody formation, I mean the complex which is formed. So this is gonna be recognized by the macrophage or the WBCs. Okay. And when the WBC recognizes this, so this will going to engulf. Okay. So the WBC is going to engulf that. Okay. And then the phagocytosis again. So thrombocytes are the platelets. Okay. So platelets, when we mention the basically the clotting is the main function over here. Okay. So what exactly does it do? So whenever there is a damaged tissue, okay, so these damaged tissues along with the platelets, so they will start releasing the clotting factors. Okay. So these clotting factors will go on activating the other things like thrombin to like prothrombin to thrombin conversion. Then this thrombin will act on the fibrinogen which was soluble earlier. So this fibrinogen will convert, or it will be converted into fibrin. Okay. So that is the insoluble one. So at the damaged part, so this fibrin which is formed, so this is going to form a mesh like structure over here. A blood plug is formed or a clot is formed, so that escape of the RBC or entry of the germ, so all that can be prevented by the clot. Okay. So now produced from the megakaryocytes, the larger cells, megakaryocytes. So when they get separated, let's say this is a megakaryocyte now. So a part of a megakaryocyte itself is a platelet. So again this is enucleated. Okay. So secretion, blood clotting or coagulation factors, which we call as the clotting factor secretion, is done. Okay. So their decrease will result in the blood loss. So you might have seen hemophilia patient. Okay. So in them there is so you might have, if not seen, okay, so you might have read about this one. So in case of hemophilia, what happens? So basically the clotting factors are absent in them. So clotting factors are not being produced, and if the clotting factors, okay, so if the clotting factors are not produced, okay, and if the clotting factors are usually absent, so in that case what is going to happen? So there is a blood loss because there is no clot formation. So continuous blood flow will be there. Okay, so this is going to form about 2 to 4 lakh per cube of a area. Okay, then platelets are derived from a megakaryocyte itself, which are again produced in the RBC, red bone marrow. Okay, so these are budded off. So I just told this is a megakaryocyte. So this is a giant cell, a bigger cell. So this is budded off. Now this part is removed, this part is removed. Okay, so budded off from the megakaryocyte itself is giving. So this is a platelet, this is a platelet. Okay, so this is also a platelet now if it is getting bud off. Yeah, so their lifespan is 3 to 10 days, destroyed mainly again in the spleen itself. So these are very important in clotting. Okay, so clotting of a blood. So at the site of injury, the platelet is going to disintegrate to release a chemical that is a thrombokinase. Okay, or a clotting factor also we can mention, so which will initiate the process of blood formation. So what we saw today, so it is about the blood. Blood is a specialized connective tissue. It has the matrix, that is plasma, then formed elements. Formed elements are RBCs, WBCs, platelets, and RBCs are having a hemoglobin. Because of that, the RBC has got a red color, or a blood has got a red color. WBC, granulocyte, and agranulocyte we saw. Okay, so the differences we saw and where they are generated, what is their function we saw. Then the decrease in thrombocyte, that is platelet, so it will result in the excessive blood loss because clot formation is not occurring. So there is a continuous blood flow. Okay, I hope this was clear for you all. Yes, so which one of the following is correct here? So serum is equal to blood plus fibrinogen, lymph is equal to the plasma RBC WBC, blood is equal to plasma RBC WBC, plasma is equal to blood minus lymphocyte. So with this only we can make out that, so it is the blood is equal to plasma RBC and WBC will be there. So now here a drop of each of the following is placed separately on our 4 slides are taken, and a drop of a blood is placed. So which of them will not coagulate? So blood serum, sample from the thoracic duct of a lymphatic system, whole blood from the pulmonary vein. Okay, and then the blood plasma. So plasma itself is having a fibrinogen. Okay, so this is also having the whole blood, means again a fibrinogen is there. So sample from thoracic, the lymphatic system, when we mention plasma is there, so again in that also fibrinogen is there. So serum is the one where the fibrinogen is absent, and if fibrinogen is not there, then there is no clotting. Okay, so that is why blood serum is the answer for this one. Okay, so which one of the following human organs is often called the graveyard of RBC? So it is the gallbladder, kidney, spleen and liver. So we discussed about this one. So this is the liver. Okay, sorry, spleen. Okay, so which of the following is not a granulocyte? Okay, so this is a lymphocyte, eosinophil, basophil, neutrophil. So not a granulocyte is a granulocyte. That means it is a lymphocyte. So this is acidic dye, this is basic dye, and it is neutral dye. So both of these are granulocyte. Lymphocyte is a granulocyte. Okay. So globulins contained in human blood plasma are primarily involved in what function? So it is globulins are proteins, that is antibodies. Okay, so antibodies are having a role in defense mechanism. Okay, so yes, so before I end the session, so let me just say about this one. So there is an extension of Prodigy where you can participate on Feb 27th. So you get one more chance to participate in this aptitude test. Okay, so all it takes is your 60 minutes of time. Okay, that is 12 to 1 p.m. and there is a chance that you can win prizes worth rupees 10 crore, where you can get the scholarship, that is a college grant you can get, and also there is a chance that you can win iPhone, MacBook, the Amazon Echo Dot, Amazon watches, so anything, you can win all of this. So you can enroll now for free. Okay, and the link you will find in the description box, and this is my referral code Vani YT to unlock the product that is there. Okay, and these are the ongoing batches. So there is a Udan batch and a Champions batch. Okay, so first of day, this is already started, so this was also started. So recordings you can watch here, and also with the recording, some classes are yet to go live, so you can attend the live classes as well. And here the difference is that the mode of delivery is Hindi here, the mode of delivery is English over here. Okay, so one shot revisions we are we are conducting, and tomorrow I have a class where I'll be conducting a quiz. Okay, so quiz on circulatory system I'm conducting, so that is at 5:15 p.m. All you need to do is download the Unacademy Learner app, and you can search for Vani Kari also. Okay, so there you will find the details of my special classes, so you can enroll for that, so you can attend this session as well. So quiz tomorrow at 5:15 p.m. on circulatory system. So we will solve about 60 questions in 60 minutes. So I'll be giving it faster. Okay, so only for 15 seconds we can conduct the poll. Okay, so you can download the Unacademy app now and try all of these features for free. So how you can do that? Download the app, open the app on open the app and click on get started, then enter your mobile number and OTP, sign up using your email ID. Okay, then you can select your class, that is class 6 to 12. So because two categories are available there, so you can select this one, and then you can choose the category ICSE. Okay, then the language in which you want to learn, so you can you can prefer to learn in English or a Hindi, so you can go for any of this one. So you can select your language, then you can use the code Vani YT to unlock the free products that are there on the Unacademy. Okay, so that is Unacademy app, you can download and start attending the free classes, and if you like them, you can go for the plus subscription, because in plus subscription the classes are taken in a regular manner, complete syllabus coverage will be done. Yes, so here thank you all. Okay, so subscribe to us, that is on Unacademy, ICSE and ISC boards, and download the Unacademy app now and start attending the free classes, the special classes. Okay. So this is gonna help you crack your finals, as we are coming up with lot of ideas related to to help you all to achieve your best with respect to your final exam, as we are conducting one shot revision, we are also conducting a quiz, then we are we also conducted this thing, that is preparation strategy, time management, answer writing hacks. So this is gonna help you all. Okay, so start taking up these free live sessions which are there on Unacademy app. Okay, so see you all in the next session. Bye bye everyone, take care, thank